Hello, and welcome back to Introduction to Genetics and Evolution. We're at a transition point in the class now. We've already gone over a lot of basic transmission genetics. We've talked about things like recombination rate, we've talked about genetic mapping, things like that. What you'll start to see now over the next couple of videos is we'll still be talking about topics related to genetics, but we'll gradually be getting significantly more evolutionary, and we'll talk about some things having to do with evolution. Starting with today's talk. Today we'll be looking at the different contributions from genetics versus the environment to various traits. Basically, how do we tell if each of the two contribute to a particular trait? How do we tell that some trait has a genetic contribution versus an environmental one? Well, the media often portrays this genetics versus environment, or nature versus nurture, as they like to say it, as some sort of raging debate. And in fact, I've had people on airplanes ask me, like, where do you stand in this debate? There is no stance. That's just ridiculous. It's nearly always somewhat of both. But we do want to be able to actually identify that there is a genetic component or an environmental component, because there are a small number of traits that only have one. Let me start with this example. This is a picture from my backyard. These are some corn plants that I was growing right next to our deck there, as you can see. Now, what I did here to make it a little bit easier to see is I put these white bars, as you can see here, just depicting the size of the corn plant next to it. So you see I have some that are about this tall, some that are about this tall, and right next to the deck I have some significantly shorter ones. So why is that? Why do you think there is this difference? Then it's not just one corn plant. There's a bunch of them that are all about the same height. A bunch of them, they're all about all the same height. A bunch of them, they're all about the same height. Do you think I split up the seeds and put all the tall corn over here, the medium corn over here, and the short corn over here? Probably not. Do you think I watered them differently? No, they're all pretty much watered the same. Well, if you think about it for a second, the answer is kind of obvious. That these corn plants out here were getting a lot more sunshine, whereas these corn plants were getting a lot less, because you can imagine as the sun was going over, they're still, all of them are still getting sun. When it gets over way over here, these ones are still getting sun, but the ones up against the deck are not getting any sun. So there clearly is some sort of environmental component to this corn. But how do we quantify this? And more fundamentally, why do people even care? Why do people want to quantify whether there's a genetic versus environmental component? Well, there's a lot of reasons. As you'll see later in the class, you'll see that we want to be able to predict evolutionary change, right? And evolutionary change requires that there's some sort of genetic component. You can't have natural selection or evolution by natural selection unless there is some hereditary basis to the traits that are being selected. Okay? This is often also used in animal and crop breeding. So let's say, for example, you're a farmer and you want to breed for corn that grows tall. And let's say that there is basically no genetic component to it. Do you want to spend a lot of energy trying to select when there's in fact going to be no response at all? Or let's say there's no environmental component. Do you want to spend a lot more resources for excellent fertilizer and things like that when it's not going to matter? They're all going to grow to the same height anyway. No, that basically you want to know this sort of information so you can spend the least amount of effort or energy possible for the best yield. This is true for both animal breeding and crop breeding. You want to know if the environment matters, and you want to know if it helps to pick parents that have particular traits. This is also very important in the context of medicine. Let's say, for example, you're a doctor and somebody comes to you and says, if my mom and dad had peptic ulcers, am I likely to get them? Basically, is there some sort of genetic predisposition to peptic ulcers? You want to be able to have an answer to these things, and this is where this kind of work is very useful. So let me emphasize again, this is not a pure dichotomy. This is very rarely an either-or question, but it's usually some of both. However, one may contribute way more than the other. So something like, for example, eye color. Yes, there's probably some small environmental component to that, but there's a very strong genetic component to eye color. That, if I could lie in the sun staring at the sun forever and my eyes would not turn blue. Something like that. Similarly, HIV status. There are some genotypes which are slightly predisposed to being less likely to getting infected with HIV. But there's obviously a huge environmental component that your behavior is very likely to affect how likely you are to get HIV. So let's dive into this. First, do genetics contribute? The way people usually identify some sort of genetic contribution, and I mentioned this in the beginning of the genetics section, is that there's some sort of parent-offspring resemblance, right? That this could indicate that genetics are important because people who are related tend to look more similar to each other than to just somebody at random from the population. However, this is not a perfect measure. And part of the reason for that, and this is something we'll come back to a little bit later on in this set of videos, but not in this video in particular, is that parents and offspring often share part of their environment too. So how do we separate these two things? How do we distinguish? Well, I'm going to show you a couple of different means. In the first part here, I'll talk about resemblance between relatives, and I'll use examples of identical versus fraternal twins, or monozygotic versus dizygotic twins as an example. And I'll use that just as an illustration, and then we'll come to the more meaty parts, which is common garden experiments or reciprocal transplant experiments. And those use the same principles as the sort of twin examples. And you'll see this in just a moment. The idea here is we want to separate genetics from environment. We want to keep one side constant and vary the other and see if there's an effect. If we're keeping the environment constant, we vary the genetics and we see an effect, then there must be some genetic contribution. If we keep the genetics constant, we vary the environment and we see some sort of effect on the phenotype, then there must be an environmental component. That's the basic principle. So looking at the two types of twins I mentioned, monozygotic, which are identical, these are ones that came from a single sperm and egg that then separated out and they shared a single placenta. These are genetically exactly the same. Okay? Dizygotic or fraternal twins are actually genetically no different from any other brother or sister. They're separate sperm and egg fertilized. You can never have identical twins that were different sexes because you can't have different sexes that have exactly the same genotype. Famous example of some twins are the Olsen twins here. They, by the way, are fraternal rather than identical, just so you know. Now, we can leverage this difference between the monozygotic and dizygotic twins. We predict if there's a genetic component to some trait. So let's say you're looking at height. Let's say there's a genetic component to the trait. The monozygotic twins should be more similar to each other in that trait, such as height, than dizygotic twins. On average, if you look across monozygotic twins, they should be very, very similar. Dizygotic might be a little bit more different because the dizygotic twins don't have exactly the same genotype. They have some different genes, whereas the monozygotic have exactly the same gene. And we're assuming these twins are basically reared in the same environment or close enough. There's no particular difference. Like if you're having kids, if you have monozygotic twins versus dizygotic twins, you probably don't treat them any differently. You treat them the same because they're your kids. So the way we measure this, the way we measure how similar or how different you are is using correlations. So using correlations and traits is that we do this. And this gives you some measure of how well traits match between groups. I know some of you may have had a statistics background, some of you may not. So for those of you who already know some statistics, I apologize for this review. Correlations are given a numeric score, which is often labeled an R. And that R ranges from -1 to 0 to +1. Now what this does is indicates how closely one predicts the other. So let's say, for example, you have a perfect positive match. That would be an R of 1. So an example of this would be your height in centimeters to your height in inches. One perfectly predicts the other, right? That you have this absolutely perfectly linear correlation between the two. Okay. In contrast, let's look at one that's not quite so strong. Here's one. Let's say you compare your height to your brother's height. You may see something more like an R of 0.7, where on average, if you have a tall brother, you're likely to be tall. On average, if you have a short brother, you're likely to be short. But it's not this perfect correlation. There are some individuals who have a tall brother but are a little bit on the short side and things like that. So it's not perfectly predictive. And again, in this case, some of this may be from genetics and some of this may be from shared environment. That's why you do see some sort of association here. Now let's go a little bit further. Let's imagine there's no correlation. You may see this in the context of your height to your neighbor's height, your next door neighbor's height. It's quite possible that you'll have some very short neighbors, you may have some very tall neighbors, but there's no reason to expect that their height is any more or less like your height. So this would be a case where the correlation is 0. Now you may be wondering, well, I've only done this range from 0 to 1. What about the negative part? Well, negative is when you have literally a negative prediction, such as daily high temperature to morning rainfall in the month of April. That you may have a lot of rainfall associated with low temperature, but as rainfall goes down, you get a higher and higher temperature. Okay, so this would be an example of a negative correlation, or a negative 0.5 in this particular example. So if that gives you an illustration of what I mean when I say correlations. So now let's come back and use this. The prediction, as I said before, is that if there's a genetic component to a trait, monozygotic twins should have a stronger correlation to that trait than dizygotic twins. Right? The monozygotic are the ones that are genetically identical, right? And the dizygotic ones have some different genes. And we're assuming that the environment is about the same in both. So what do we see with the data? Well, if we look at correlations with twins, we often see this difference between monozygotic twins and dizygotic twins. Here are a couple of examples. One is for IQ, or the supposed intelligence coefficient, that monozygotic twins tend to score very similarly on these tests. The correlation is 0.85, whereas dizygotic twins have a correlation of 0.42. This suggests there is a genetic component to what is measured here as IQ. Same sort of thing for some diseases. When we look at gastroesophageal reflux disease, monozygotic twins have a 0.29, dizygotic twins have a 0.13. Again, we see this higher association when they're genetically the same than when they're genetically similar but not identical, which means that there is some sort of genetic component. There may be an environmental component. I should stress this. None of this proved the absence of an environmental component. All it shows is the presence of a genetic component. I should stress that. Okay? Now, since this didn't prove the absence of an environmental component, how do we look at that? Well, what we can do there is keep the genetic relations constant but vary the environment. What you do is you look at monozygotic twins reared together versus monozygotic twins that were reared apart. And your prediction is that monozygotic twins reared together should have a higher correlation in these traits than monozygotic twins reared apart if there's an environmental component. The reason is those monozygotic twins reared together have exactly the same genes and the same environment, or very, very, very similar environment. Whereas monozygotic twins reared apart have exactly the same genes, but somewhat of a different environment. Now, this has been done quite a bit, especially in the context of psychology, but in other areas as well. So here's some results. Looking at the body mass index, or BMI, monozygotic twins reared together have a correlation of 0.74. In contrast, monozygotic twins reared apart have a correlation of 0.70. This suggests there may be some environmental component, but it is surprisingly very small. Very small, surprisingly small environmental component. You may think this is unexpected for something like body mass index, but in fact, for this particular study, that's what they found. In contrast, for verbal ability, monozygotic twins reared together had a correlation of 0.76, whereas monozygotic twins reared apart had 0.51. So it suggests a very strong environmental component to verbal ability. Now we can again do the same thing with the monozygotic versus dizygotic. So looking at this showed a strong environmental component. This also shows a strong genetic component because again, the monozygotic together versus dizygotic together means the environment's the same. Mono versus di is changing the genetics. In this case, we see a pretty large genetic component and a moderately large environmental component for the same trait. Again, seeing this difference doesn't necessarily mean that it doesn't mean the absence of a genetics component. It just means the presence of an environmental one. Okay? So again, what we try to do is we try to control for environment and vary genetics and see if there's an effect, or control for genetics and vary the environment and see if there's an effect. Well, the same principles are done when you're looking at animals or plants and things like that, and not involving mono and dizygotic twins. And in fact, these have been done for quite some time. Let me show you some classic work. This is from the 1930s, work done by Clausen, Keck, and Heisey. And they were interested in these differences among plant forms identified from different elevations. So they got some plant forms from, as you can see here on this elevational gradient, from timberline, which is very high in the Sierra Nevada of California, from Mather, which is moderately high in elevation, and from Stanford, which has a very low elevation, just above sea level. And what they found is that the plants, these are the same species, looked very different, these different forms. At the high elevation, they tended to be kind of thin, sparse, not particularly bushy. Here's a color photograph of the high elevation form. As you got to lower elevations, you got this much bigger, bushier form down there. Is it genetics or is it environment? Well, they pioneered a set of work that was extraordinarily influential. So one thing they tried were these common garden experiments. These had actually been done by other researchers in the past, but they applied this to these particular plants in California. Essentially what they do for this is they grow plants obtained from different places in a single environment. So the idea here is they're trying to keep the environment constant, but getting these plants from different places, these likely have different genes, right? Because they're coming from different places. They likely have a different genetic makeup. So if the form difference between, say, the high elevation plants and the low elevation plants was all environmental, right? If you grow them in the same place, they should all look the same. In contrast, if this form difference between the tall and short plants was all genetic, then the plants should look quite different. Even if it's partially genetic, they should look quite different. This is very similar to the concept of mono versus dizygotic twin studies because, again, we're using unrelated plants, so they have different genes, but looking at them in a constant environment. Well, what did they see? In fact, they found there was a form difference apparent even when grown in the same environment. So these are ones from the species Potentilla gracilis, and grown in one spot in Mather. So this is that intermediate elevation spot. They found quite a big difference in form. And this suggests this form difference does have a genetic component. It does not prove there was no environmental component. It only proves there is a genetic component. Okay? Well, how do we do the other half of this? Well, the other thing you can try is what's referred to as a reciprocal transplant experiment. In this case, you grow plant forms obtained from a single environment. So since they're from the same place, we're assuming the genetics is fairly constant. And you grow them in several different environments. So you're taking all these ones that have the same form, and you're growing them in different environments. So maybe you take it from Mather, grow it in Timberline, in Mather, and in Stanford, for example. If part of the form difference is environmental, then they should in fact look different. In fact, if the form difference was purely genetic, then all the plants would still look exactly the same, right? Because they're all derived from that same place. Okay? And this is the same concept as these twins reared together versus apart, that we're using the same genetic makeup, right? And we're varying the environment in which the plants are grown. And in fact, with this, they did still see an effect. The form difference for the same type grown in different environments was still apparent. This is showing Potentilla glandulosa. And this was grown at Stanford, Mather, and Timberline. And you note again, the Timberline ones are quite small. The Stanford ones are much bigger and bushier. This suggests this form difference does have an environmental component. Now, everything I've shown you so far is how to tell, is there a genetic component? Yes or no. Is there an environmental component? Yes or no. And remember, just because you say yes to one does not mean you're saying no to the other. Because quite often, as you saw with these examples, there is some of both. Now, what you probably want to do is to quantify how much of the variation that you see is genetic versus environmental. Well, that we'll come to in the next video. Thank you.